I hope you all would be good. And now we go further into the detail of this disease and the immunity chapter that we have developed in the body. If I were to be saying or recalling something from the previous class, so over there we have looked at our human body has developed some specialized kind of the physical and the chemical barriers, and they would not allow the pathogens to get an entry into your body. But the situation is that in some cases, these mechanical and the chemical barriers that we have caught in the body, so they fail to restrict the pathogen onto the surface of the body. So the pathogen literally would get into the cells or near to the cells of your body. And over there, the ability of the body is now going to take place, and that would be the immunity. And by the word immunity, what we mean is that this is the body's ability to fight against the diseases once they have entered into the body. And in the immunity, what we understand is that my body would be initiating a kind of a response, and this response is entitled as immune response. And how we define this immune response, that this is the reaction that would be started or it would be initiated in the body against the foreign pathogens. And we will see in a while that these foreign pathogens that are getting an entry into the body. So on their surface, they have these special proteins present. They are referred to as antigens. And against the antigens, our white blood cells that we have in the body, so they would be activated or they would be identifying them and binding with them. So, in terms of the immune responses that my body would be initiated, so we have got two type of the immune responses. The first one would be entitled as a primary immune response, and the second immune response that my body would be triggering. So it is entitled as a secondary immune response. What primary immune response is that this would be carried on or this would be started or initiated in the body whenever some pathogen it gets an entry into the body for the very first time. Like as it happened in the case of the COVID, that anyone who was getting infected by the COVID, so they were taking some time to become immune against it or to become protected against it. So what was the reason? That our human body was initiating the primary immune response. The person, those who got recovered from the COVID, so later when they developed the COVID or they got the COVID, so they recovered easier because now when the COVID was attacking our body for the second time, so my body or your body would be initiating the secondary immune response. So the primary immune response happened first when the pathogen would enter for the first time, and the secondary immune response would be initiated or the started whenever the pathogen or the same pathogen when they would get an entry into the body for the second time. The idea is that these immune responses or internal immune reaction, this is provided by the type of the white blood cells that we have with us, and these are the white blood cells. So initially, our concept, our idea of these white blood cells should be very much clear, so that we should be clear about what immune responses actually are. So among the white blood cells, we have two major categories. We have these phagocytes coming, and then we have these lymphocytes coming. Among the phagocytes, we also have got two further distinct categories of these phagocytes, and they are the neutrophils and the macrophages. But differentiation is not that much required for your course, only the word phagocyte should always be there in your mind. And when we say phagocytes, so they would eat up the pathogens. And when we talk about the lymphocytes, so they produce some proteins out of them. They are the antibodies, and they would also keep a track or the memory of the pathogen. They would remember the pathogen so that our body is in the position to be initiating this secondary immune response. So if I be uh, taking you toward this, so as I've said, that the white blood cells, they are the phagocytes and we have the lymphocytes. In the phagocytes, we have these neutrophils and the macrophages. And then in the lymphocytes, we have these plasma cells and we have this memory cells. The plasma cells is not a part of your course, but still for the higher levels, you must know that these are the type of the lymphocyte that secretes the antibodies. So our idea initially would be that how come these white blood cells that initiate the internal immune response, so they would appear under the microscope. So if I were to take you to this, So you see that on this side we have got these phagocytes with us, and here we have the lymphocytes. If you were to identify a neutrophil among the phagocytes, so the idea is that their nucleus is multiple lobed nucleus, and they have got these intracytoplasmic granules in them. So whenever you see such structures given in the exam, so you would say this is a phagocyte. Likewise, if you were to come across a diagram where you will have the cell membrane being shown and the nucleus of the cell is shown as an oval nucleus, so again, this is a macrophage, but generally in the exam you could write it as a phagocyte. Now, the difference that we have to see in case of these lymphocytes that we have with us, so you see that they also have the nucleus. This is neither oval shape nor multiple lobe nucleus. But rather than this nucleus is covering most of the part of the cytoplasm. And another interesting thing about these lymphocytes is that in their cell surface membrane, they have developed these protein receptors, and these protein receptors would allow them to differentiate between the normal cells and the pathogens, those who come in our body with an intention to be causing the diseases. So if in the microscope or if in the diagram these cells are given, so whenever you come across the multiple lobe nucleus, you have to call this as a phagocyte. Whenever you would have a cell and its nucleus would be coming, and if this appears oval shaped, so you would again call this as a phagocyte. Lymphocytes overall, they also have a nucleus, but in size, if you see, they are drawn smaller than these phagocytes, and you always have to look on their nucleus. If nucleus takes up most of the part of the cytoplasm, so this would be the lymphocyte with us. Now, the idea is that, okay, if the pathogen has entered into my body and the white blood cells, they have to react against it, or they have to initiate an immune response against them. So how come our white blood cells would be in the position to be distinguishing between or to be differentiating between that whether this is the normal cell of the body or if this is a pathogen with us? The idea is that these pathogens that are entering into the body, like it could be the bacteria or the fungus or the protoctist, so on their surface, we have got the presence of this protein in them, and this protein is referred to as antigen. So overall, this is a pathogen. And on their surface, the proteins that we have got, they are referred to as the antigens, and they are the non-self antigens because they are not present on the surface of our body cells, but rather than on the surface of the pathogens. And to these antigens, when the white blood cells, they would bind with them. So ultimately, the immune response would be initiated. On the other way around, the situation is that my normal cells of the body, they also have got some protein receptors on them. Like if I were to take you to an example of the red blood cells. So the red blood cells, they look as a biconcave disc. So you just need to know is that this is not the nucleus, but rather than this is the pinching that we have got at the center of the red blood cells. So these red blood cells, they also have got these, you could say, protein receptors on them. But against these protein receptors, the white blood cells, they would not have something complementary to them. Okay, so the normal red blood cells, they also have these surface proteins. They are the antigens again, but they are the self antigens. Self antigens mean that they are the proteins of the normal cells, and against these receptors or against these self antigens that we have on the surface of our normal cells, there would be no white blood cells that would bind with them. So in this way, our white blood cells, they have an ability to be distinguishing between the pathogens that we have and the normal blood cells of the body that we have. And at any cost, we always have to ensure that my white blood cells must never attack the normal cells of the body, because if they would start attacking the normal cells of the body, so this can even take a person into the autoimmune diseases. Now, moving onward, as I have said that among the white blood cells, we have these phagocytes coming. So the idea is that how come these phagocytes, they would be able to be removing the pathogens out of the body. So what we say is that these phagocytes, they are scavengers. By the scavengers, what we mean is that they literally eat up the pathogen that has entered into a body. So from the word phagocyte, what you must remember is the word phagocytosis. And the phagocytosis is that mechanism in which any of the pathogen that has entered into my body, so it would directly be ingested or it would be eaten up. The it would be eaten up by the phagocytes. And the other idea is that these phagocytes that we have got in our body, so they provide non-specific immune response. By the word non-specific immune response, what we mean is that irrespective of whether the pathogen is the fungus or the bacteria or the protoctus or the virus, so whatsoever the pathogen having a non-self antigen on the surface, when this would enter into the body, these phagocytes would say that, okay, fine, you have come now. Let us have a party, and these phagocytes would start eating these pathogens up. So we must be clear about it. How come this mechanism of the phagocytosis would be brought about by? So now if you see that I have got this diagram for you. So this is the diagram of a phagocyte, and on the phagocytes we have got some protein receptors. Okay, these protein receptors can have multiple shapes, so they can bind with any of the pathogen that would get an entry into the body. Now the other thing is that at any cost I also have to ensure is that while these phagocytes that we have and they are going to be digesting or when they are going to be ingesting the pathogen, so they themselves must not die. Only the pathogen must die. So first idea is that any of the pathogen that has entered on the inside of my body, so these pathogens, what they would initially do, that they would make an attachment with the cell surface membrane of this phagocyte that I have got. And this attachment is attributed to the fact that these phagocytes, they have the protein receptors on them, and these protein receptors, they would bind with this pathogen that have come on the inside of the body. The other situation is that since I want to save the phagocyte itself, so the next step would be that this pathogen would be enclosed inside a vacuole. Like if you were to see this, that after this pathogen has made the attachment with the cell membrane of the phagocyte, in the second step, what is happening is that these phagocytes, they enfold their cell membranes inward. So the pathogen is enclosed within a sac. And here you see that we have got a sac made now, and the sac has got the pathogen in it. The other thing is that these phagocytes in their cytoplasm, they have got these vacuoles in them. And these vacuoles, they have some specialized enzymes coming in them, and these are the hydrolytic enzymes. So if I were to open up this vacuole directly into the cytoplasm, so the enzymes, they would come into the cytoplasm and they would kill this phagocyte as well, which is what we don't want. So what we have done is that we have enclosed a pathogen in this sac, and now these vacuoles, those who have hydrolytic enzymes in them, so they would fuse with the membrane of this sac in which the pathogen has been enclosed, and now the digestive enzymes would be released out of them. And when the digestive enzymes would be released out of them, so as a result of that, this pathogen would be completely digested and no harm has been done to the phagocyte that we have on the inside of the body. So if I were to quickly summarize this, the pathogen would enter and this would make a contact with the cell membrane of the phagocyte. Then the cell membrane would enfold inward and an enclosed sac would be found. To this enclosed sac in which we have this pathogen present, so the vacuoles that have hydrolytic enzymes in them, they would fuse and they would release these enzymes, and by that the pathogen would be digested. So I hope you would be clear about that what immune response is, how many type of the immune responses we have with us, what white blood cells are, how they appear under the microscope, and how come the phagocytes, they would go onward with the mechanism of this phagocytosis we have got. So see you in the next class.